For this video, we're going to be talking about variations on Mendel's laws. So incomplete dominance results in intermediate phenotypes. So Mendel's P crosses always look like one of the parents or parental varieties. One of the crosses, one of the offspring of the parental crosses always look like the parents. And so this is called complete dominance. So for some characteristics though, the appearance of the F1 hybrids falls in between those two phenotypes of the parents. And so this is called incomplete dominance, in which neither allele is dominant over the other, and then the offspring the offspring will have an expression of both of the alleles. And this is very beautifully explained in flower color. So if you was to take from the parent generation a red flower and a white snapdragon, for example, and you cross them, you're going to get an individual who has a pink color. So you have complete dominance here of the colors in each individual to a hybrid individual that is now expressing both colors, which this creates incomplete dominance. So if you are to cross two hybrid individuals here that are pink, you're going to get a test cross of individuals that now express, have probabilities for having all of the flower colors, both parental and the F1 generation colors. So incomplete dominance does not support the blending hypothesis, which was debunked because the original parental phenotypes reappear in the F2 generation. So one example of incomplete dominance in humans is hypercholesterolemia, in which dangerously high levels of cholesterol occur in the blood. And if you're heterozygous for this, means you have intermediate high levels of cholesterol, but they're not dangerous. So if you're homozygous for this trait, homozygous dominant, you will produce enough LDL receptors. LDL is low density lipids in the blood, which is bad. You don't want LDL, high LDL concentrations in your blood. And so normally you will have LDL receptors that can grab onto those. If you're heterozygous here, it means that you have some that can attach, but some are still left in the blood, which is not good, high cholesterol levels. But if you're homozygous for this hypercholesterolemia, you don't have those LDL receptors, and so you have high, severe levels of cholesterol in the blood, which is very dangerous. So many genes have more than two alleles in a population. Although an individual can carry at most only two alleles, one you inherit from your mother, one you inherit from your father, there can be multiple alleles for a trait in a population, in the overall population. So an example of that would be your human blood, the ABO groups. There are 3 alleles for blood that exist for humans. So the 4 human blood groups, AB, AB, and O, result from a combination of these 3 different alleles. And so A-type and B-type alleles are both expressed in heterozygous individuals, which is a condition known as codominance. So in codominance, neither allele is dominant over the other, remember? And so you have an expression of both alleles that are observed in the phenotype of the heterozygous individual. So AB blood is also an example of a codominance type. So this is an overall summary of your blood, the 4 different types of blood. So let's focus on these individually. So for type A, you carry either 2 dominant A alleles, or you have 1 dominant A allele and you're carrying 1 recessive allele, which happens to be type O. So it means these individuals here are going to create carbohydrate A on their red blood cells. Type B blood carries either two dominant B alleles or one B allele, and then one recessive allele, which is going to be for type O blood. But they are now type B. These individuals would carry carbohydrate Bs on their red blood cells. Then there's type AB individuals would carry one type A, one type B dominant allele. And so these blood types carry both the carbohydrate A and carbohydrate B um, on their red blood cells. And then there is type O, an individual who has inherited two recessive alleles, and so they have no carbohydrates on their blood. So how does this affect your blood and when you're having to receive blood, for example? So if you are type blood A, it means you have anti-B antibodies in your blood. So if you were to receive type O blood from someone, your blood cells would be fine. If you were to receive type A, you would be fine. But if you were to receive type B or type AB, because there's B present and you have anti-B antibodies in your blood, your blood would clot or clump, and so that's not good, right? So if you're a type A individual, you can only receive type O and type A blood. Same is true for type B, in that you have an anti-A antibodies in your blood. So you can only receive type O and type B blood. Where if you were to receive anything with an A, so type A or type AB, that blood would clump and it would harm the individual. Now, type AB blood has no antibodies, meaning that they can receive blood from anyone. So type AB is called the universal receptor. If you have type AB blood, you can receive blood from anyone, which is actually a very good thing if you ever needed blood. Type O, though, has both the anti-A and anti-B antibodies, meaning they can only receive type O blood. Because type O is recessive, they can only receive type O blood. So type O is called the universal donor, meaning they can donate blood to anyone, but they can only get type O back for themselves. Another example of where a single gene may affect phenotypic characters is in pleiotropy. Pleiotropy occurs when one gene influences many characteristics, or it has a cascading effect on the characteristics of a certain part of the body. For example, sickle cell disease is a human example of pleiotropy, where the disease affects the type of hemoglobin produced and the shape of the red blood cells. It can also cause anemia and organ damage. And then sickle cell and non-sickle cell alleles are codominant, which means that it can be in heterozygous individuals. And the carriers of sickle cell disease are resistant to malaria. So this one trait, this one gene trait, can have all these different effects on the body. And so this is just an example of you have normal white blood cells, and then you have those that are sickle-shaped, which are abnormally shaped compared to the normal red blood shape. So an individual that's homozygous for sickle cell can produce sickle cell hemoglobin, which is abnormal. And it can crystallize, causing red blood cells to become sickle-shaped. And so these sickle-shaped cells can actually cause a lot of problems. It can damage your organs, leading either to kidney failure, heart failure, spleen damage, brain damage, many different other effects such as pain and fever, joint problems, pain in your extremities, like your fingertips, your nose, physical weakness, anemia, because the hemoglobin has been affected in these cells and they're not able to transfer oxygen throughout the body like normal red blood cells would do. And because of their odd shape, many times they get stuck or jammed in your capillaries, which are your blood vessels on the very extremes of your body, like your fingertips, which creates very painful side effects. So sometimes characteristics can result from polygenic inheritance, in which you can have a single phenotype be the result of many genes. Human skin color is an example of a polygenic inheritance. And so I like to think of this as these are additive traits. So an example would be, or at least the best example would be color. Anything to do with color. So skin color, for example, is what we're going to be looking at. So in a parental generation, the P generation, you can have an individual that has very light colored skin compared to an individual with very dark skin. And you can see here, the dots here represents the multiple genes that this individual has and the traits that they seem to code for. When you cross these two individuals, you're going to create the F1 generation that are considered hybrids. And you can see that these hybrids have a skin color that is an intermediate between the very light and the very dark. And they have an equal number of light versus dark traits or genes. And they are true heterozygous for those genes. Now, if used across two heterozygous individuals, which this occurs all the time, you can produce a very complicated, because it's not a dihybrid, it's a, I guess the word would be like a trihybrid cross, where you're crossing three different traits. And you can see here, you're going from very light skin all the way down here to very dark skin. And so the numbers of individuals with the different skin color types are shown here, where you would have one individual that's very light skinned compared to one individual that's very dark skinned, and then slowly a mix of the two with higher probabilities towards the middle. And so this is just to show you that skin color is polygenic in that it relies on multiple genes that you inherit. I think if I'm correct, I think skin color is actually 8 genes. It's an 8-gene trait. And so if you are to cross an individual, if you've ever was to wonder, why do some individuals, say in your family, in your own in nuclear family, why are some of your siblings, for example, darker or lighter than you? Or is there someone in your immediate family that is much darker compared to everyone else or much lighter? Well, this is why, because these genes are additive. So depending on if you get more that are light colored or more that are dark colored, then you're going to have a skin color that represents that additive effect. So skin color is polygenic. Another example would be height. So height is it works the exact same. So if your parents are very tall, taller individuals, then their offspring are more likely going to be very tall. But it's not unheard of if one of them was to actually be short. These are additive traits. It all depends on the probability and which ones that you inherit. And this is just to show you the differences in height in a close nuclear family here. So the environment affects many different characteristics as well. So many characters result from a combination of heredity and the environment. For example, you can inherit many dark skin color traits, but your skin color is affected by exposure to sunlight. Um, Susceptibility to diseases such as cancer um, is also environmentally stimulated. And then sometimes identical twins can show some differences. So where if they actually are raised or prefer different environments, they can actually start to show some differences. So only genetic influences are inherited, but the environment can affect your phenotypes to a degree. And this is just showing you how you can have different skin colors based on just light exposure. And so these are some examples of twins in that even though they're very similar, they can be very different. Where you can see here, one prefers seems sunlight more than the other.